we did have our other poll question. If we can show the results of that, Miles, and we'll jump into the next half of our discussion. So it looks like IVT raw materials have taken the cake as where there have been the hardest supply, right, of cGMP raw material, followed by, it looks like, lipids, or no. People have been lucky. And then it looks like it's followed by plasmids and buffers and chemicals. So I would kind of, we can address the poll question, I think, in my next question to you guys. And Greg, we can start with you. Are there, based off of this sort of list, right, in this poll question of materials that we're working with, are you finding that, we're near I hate this word, commoditization, right, for any of them in terms of the quality, availability, et cetera, of these materials? And, if we're not near for any of them, where are you finding the greatest variability in terms of availability and also quality?
Yeah, great question. And maybe I'm a little surprised to see IVT raw materials, but that's a big bucket. My guess is that enzymes is probably driving that, because to me, especially unmodified nucleotides and buffers and general reagents and consumables, which is all part of your IVT, is the most commoditized thing. Maybe we can argue some of the cholesterol, very commonly used excipients, are also on that commoditized platform. The things that are furthest away, so again, not surprised to see lipids high on that, because the ionizable lipid especially, and some of these newer lipid alternatives, whether it's a PEG alternative or a lipid with some targeting ligand, is very custom to a product and almost as treated in terms of customization as an API. So I think we're furthest away from commoditization, if you want to use that word there. So again, I do agree with the list that custom enzymes, next generation enzymes, and lipids, and pDNA. I would just say we've come a really long way as an industry for linearized pDNA, linearized purified pDNA as a platform. But of course, every individual pDNA has its nuance, and particularly when you get to larger and larger things like self-amplified or pDNAs for other templates, there is some customization. So that's in between.
April, what do you think? Are you kind of surprised in the same way as Greg is with IVT raw materials? Are there areas that you're also facing similar constrictions, or you see sort of more of a push, right, for that customization versus we can just buy, right? We can just buy it off the shelf.
I would make the same assumption that Greg did, that the IVT roadblock is the enzymes. I mean, a couple years ago, it was tough to even source all the enzymes that were needed to an appropriate grade. Meaning that wherever you're getting it from is well controlled. I mean, sometimes they call it GMP, sometimes they don't, but the point is that it's made in a well controlled manner that you can qualify.
GMP is not actually a grade.
I mean, not for incoming raw materials, but it is important to have control of those, and the industry was not there as early as a couple years ago. So, and I think it's still, if you're going after what they call the GMP grade reagents or the enzymes, that could probably still be tough to source because everybody's after it. I don't think it's probably, I would assume, a lead time issue rather than a quality issue at this point. But everybody, like, no matter what industry you're in right now, there's intermittent issues with buffers, with resins. That's the thing. Plasmids, I think the RNA manufacturers are getting a lot better about having either in-house plasmid production themselves, or they've established preferential partnerships with the plasmid suppliers so that they can winnow down those lead times for plasmids. But there can still be issues, I think, if you're encoding your poly A tail into your plasmid, making sure you have a manufacturer who understands how to monitor and make sure that you're not, because those AT-rich regions on plasmids tend to get a little bit snipped, or they'll reduce in size if you don't watch them. So making sure your plasmid manufacturer understands that has the right sort of plasmid construct, or you do, to stabilize that. It's pretty important. You don't want to be losing your poly-A-tail later. So
that's a really good point. Yeah.
The LNPs are the big barrier to me, because you know, they are all, that's where it's another IP minefield issue, where everybody's after their proprietary formulation that usually includes some sort of customized or specialized lipid. That's how they differentiate it. So once you manage to get hold of your own IP or bought somebody else's, you got to figure out how to make that proprietary lipid. And a lot of times when they're first made, they're made research grade in a very, very uncontrolled way. And then everybody runs off and does preclinical studies with it. And, you know, as a tech ops person, like when I'm facing, the lipid's coming along, and not to be standards yet, and I don't know what it was. So you often have to mess with synthesis pathways to, like, even in a lipid synthesis pathway, the precursors can be limited in supply. So sometimes you have to switch the synthesis pathway itself between research grade supply to GMP supply, so that you can scale up and have consistent synthesis available. That changes the whole profile of the lipid. You have like, they are novel excipients, if they're new and proprietary. That's a whole nother minefield too, where you've got to not only confirm that the product you're making with it isn't toxic, you now have to treat the lipid as an excipient, which has a probably dodgy history because it was made research grade and transferred. You can backbridge, and I don't know. So it can be quite tough dealing with these proprietary lipids, I think. But once you've got one locked into a consistent process, you've managed to bridge back to your original preclinical studies, and you manage to get it through the excipient tox qualification, where you have to, like, do genotox with all the animals models and make sure it doesn't do dodgy things there. Then you're good to go. But it can be a bit of a long road.
You had fantastic points, April. I love that you're focusing on that.
So many
customizable lipids, which are, as we said before, in many ways treated like APIs. But as you say, where some of the API analogy breaks down, some of these are so lipophilic and so challenging to manufacture, which is true certainly for some APIs, that the purification steps are, really very hard to move away from chromatography on some of these synthesis. Some of them are designed to be biodegradable, so you're really dealing with some CMC challenges there, and it brings you all the way back to the first comments that we had about understanding the impact of your material attributes on your final product, right? What does that degradation profile and what do those impurities mean to your LNP synthesis, to your mRNA performance in the final product? We're all familiar also with the propensity for some of these small molecules to form adducts with mRNA that can render it untranslatable. So all of this needs to be really looked at closely. Great.
And thinking forward to, like, how it's going to be managed in the facility too. Like, I had a lipid just dead end for me once because, in a research setting, it was dissolved in ethanol. And when you scale that up to, like, what would actually be required in a manufacturing space, the volume of ethanol was way beyond what the facility could safely handle. So we had to, lose a few months there going back and figuring out, well, how do we solubilize this without 100 liters of ethanol? It was heated, so a really high temperature. Just thinking about this stuff proactively in advance really helps.
No, excellent point. And just to, you asked me in the very beginning, Anna, where the industry is from. For LNP manufacture itself, most processes do require some level of ethanol, and when you get to large scale, that requires explosion proof for ATEX handling. And because most of these are injectables, you also need a grade C environment. You are producing a sterile product. Much of it can be still upstream of the aseptic window, but you're still in grade C. So that combination of grade C manufacturing facility with ethanol handling explosion is very rare. It has definitely evolved. It has definitely become much more common, driven by the LNP. I mean, there were much fewer products that needed this this combination. So that is another area in terms of supply chain, as April points out, the LNP production